Hello everyone. Today I'm sitting here with Dr. Judy Campisi, who is a professor of gerontology at the Buck Institute for Research on Aging. Judy has made significant contributions to the field of aging, particularly for her research on cellular senescence, which I'm sure we're going to talk quite a bit about today. I've personally been following Judy's work for several years, so I'm very excited to be able to have the opportunity to have discussion with her. So, Judy,
Thank you.
I'm ready to just jump in and talk about all things aging. You know, most people when they think about aging, they think about the passage of time, a chronological number, but there's a lot of molecular changes that occur with aging.
Yeah. Well, it's a biological process, and it has its roots in evolution. It has its roots in actually gestation, but it is not just the passage of time, for sure.
So, can you maybe talk about some of the molecular changes that do occur that can contribute to the aging process?
It's a good question. You know, right now, I think it's safe to say that most people who work on aging believe there are a few fundamental molecular and cellular processes that drive aging in multiple tissues, which is why all of the diseases of aging, even though they're from very different tissues, all begin to rise exponentially after maybe 50, 60 years of age. But the truth is, we don't know exactly how many processes there are, and we don't really know precisely in each tissue what the major driver is. So there are theories, the free radical theory of aging, loss of mitochondrial function, the accumulation of senescent cells, which I'm sure we'll talk about more. But for now, I think there are two things we can say: that these fundamental processes almost certainly are driving tissue health. As for what actually drives lifespan, we really don't know.
Yeah, I guess that's an entirely different topic, I mean, to some degree.
I mean, they're related. Nobody dies of good health.
Right.
But the truth is that we don't really understand, for example, why a mouse lives 3 years and a human lives over 100 years, I mean, if you're lucky, right. And what it is that evolution had to do to take these two organisms, which are genetically pretty similar, but have a 30-fold difference in lifespan.
Right, yeah. Just kind of on the same topic, did you ever see that paper that was published? Maybe it was like 2015, I believe. It was out of Japan. And the researchers had looked at a variety of different cohorts of aging individuals. So they looked at people that were considered elderly. So these were like 80-plus-year-olds. Mm-hmm. They looked at centenarians. They looked at the semi-supercentenarians and then the supercentenarians.
Right.
And they looked at a variety of different biomarkers. I believe it was something like 12 different biomarkers. They were looking at immunosenescence in the immune system. But they were looking at telomere length. They looked at blood glucose levels, insulin sensitivity, inflammation markers, and kidney function, like all sorts of things in each, age
cohort,
group. Yeah, in each cohort. And what was found, sort of to my surprise, but not too much to my surprise, is that the only, biomarker that was indicative of successful longevity in every cohort was suppression of inflammation.
Yeah, I did read that. I did read that, yeah.
Whereas like telomere length affected if you could live to be, I don't know if it was 100 or the 80 group, one of the earlier groups. But so that was kind of interesting because it sort of, to me, this whole idea of inflammation, which causes...
Or inflammaging, as Claudio Franceschi. No, this is the term that was coined by Claudio Franceschi in Italy. Yeah, and it really refers to the fact that, well, if a pathologist were to take a liver sample, say from a 15-year-old and a 50-year-old, he or she could probably instantaneously tell you who was young and who was old. Mm-hmm. One would be just looking at the structure of the tissue, but the other is he or she would look for what we call a low-level sterile chronic inflammation, meaning low-level infiltration of some immune cells, but no evidence of a pathogen. And so this is a characteristic of almost all aging tissues, is this low-level inflammation.
Most people don't really, I think they hear the term inflammation. They don't quite know what it means.
Yeah. So there are two important, well, actually, there are two major modes of inflammation, right? So one is acute inflammation. So you cut yourself, turns bright red, that takes 24 hours. And you'll die without that. I mean, that's the acute response of your innate immune system that's going to try to protect you from infection, but also start the healing process. That acute inflammatory response has to die down after a few days because it's destructive. The innate immune system isn't very intelligent. It's designed to kill non-specifically. So these innate immune cells make hydrogen peroxide, nitric oxide, bleach.
Right, hypochlorite.
Exactly. And the whole idea is just to kill non-specifically until your adaptive immune system can figure out what precisely needs to be killed. So that's your T cells and antibody response. But that takes a few days. So you need both. Low-level chronic inflammation or sterile inflammation is similar to acute inflammation in that many of the players are the same, but of course it's at a much lower level. If it were that fulminant, you would probably die very quickly because your tissues would fall apart. But nonetheless, with aging, there's this constant increase in these innate immune cells that infiltrate your tissue. And after a while, the tissue does begin to degrade and lose its structure and lose its function.
Yeah. So it's not only, affecting the tissue, it's affecting, DNA, it's affecting...
That's right, because these are damaging agents. And so it becomes like a feedback loop. You have, cells that are being exposed to these mostly oxidants, and yet then DNA gets damaged, and then the cell responds to the DNA damage, and it makes things worse, and you get into these feedback loops. So the question is, what is the cause of that low-level chronic inflammation? Of course, the short answer is we don't really know, but we have some clues. We have some ideas. Some people have argued that with age, the barrier in your gut breaks down slightly, not enough to cause bacteria to invade your tissues, but enough to enable bacterial products to leak into your bloodstream. And then, of course, your body is going to respond and go, oh my goodness, is this an infection? And so you get this slow buildup of immune responses. The other hypothesis, major hypothesis, is that damaged cells, which are what we now think are mostly senescent cells, produce as part of their response to the damage cytokines, which then attract the immune system. And those are not mutually exclusive. It's probably both. Right,
absolutely. Can you explain to people who are listening and watching, what exactly a senescent cell is and how, it's obviously, you just mentioned, it's producing more of these inflammatory cytokines, and which to me sounds like it accelerates the aging process. But
yeah.
Sort of just, what is a senescent cell? Why do they form?
Yes, very good question. So, we meaning the field now, I think, would agree that senescence can best be understood as a stress response. It's the way cells have evolved a program to respond to certain types of stressors. And the result of that response, that stress response, is twofold. The first is that cells will lose the ability to divide, essentially irreversibly. So what happens is tissues begin to accumulate stressed cells that can no longer divide. The curious thing is that they also tend not to die, or they don't very easily die. So they accumulate with age, very slowly and gradually. They're always at pretty low numbers, but they go up with age. The other part of that stress response is the cells turn on a program that causes them to secrete molecules that we classify as being bioactive. And the reason why that term is vague is because there are probably 50, 60, maybe 70 molecules that these cells begin to secrete. And so they have many, many activities. Some of them attract the immune system, so they're cytokines and chemokines that are immune attractants. Some of them are growth factors that will alert the neighboring cells, because maybe now, if you have a need for proliferation, the senescent cell itself cannot divide, but it may want to tell its neighbors, hey, start dividing. They secrete proteases that remodel the tissue. That would also help with regeneration and repair. And they also, now we know, very new data from our lab, they secrete bioactive lipids like prostaglandins and leukotrienes, which are very important for modulating inflammation, fibrosis, but also, again, tissue repair. Mm-hmm. So the way we now think of the stress response is that it's a double-edged sword. There's tons of data that go back 2 decades or more, showing that the arrest of cell proliferation is extremely important for preventing cancer. So that stressed cell is in danger of becoming a cancerous cell, and the stress response intrinsic to the cell says, no way, you will never divide again, and therefore you cannot form a tumor. So there are mouse models now, and even some people with mutations in the genes that regulate that growth arrest. And those people die in early death due to cancer. And the mice die in early death due to cancer. So we know the growth arrest suppresses tumorigenesis. The secreted factors we also now know can be beneficial for tissue repair and wound healing. So we've shown, for example, in the skin, senescent cells appear at the wound, and they produce growth factors that help the wound heal. That's the good news.
Yes.
The bad news is, as these cells gradually accumulate with age and don't disappear, they begin to drive this process of chronic inflammation. And that chronic inflammation caused by senescent cells is also a double-edged sword. On the one hand, it attracts the immune cells that will eventually begin to cause tissue destruction and degeneration. On the other hand, the cytokines that attract the immune cells can also have an effect on neighboring cells and cause them to not function properly. So, for example, some of the cytokines that senescent cells produce cause what's called an epithelial-to-mesenchymal transition. So what is that? Most of the organs in your body are composed of epithelial cells, and these are cells that absolutely must talk to each other. Mesenchymal cells are the support cells, like in the skin, the epithelial cells are the outer layer. The support cells are in the dermis. Those are the mesenchymal cells. They don't need to talk to each other. They need to signal. They're basically telling the epithelium what to do. Now, when an epithelial cell becomes more mesenchyme-like, it stops talking to its neighbors. And that means the tissue is going to start losing function. And so senescent cells can change epithelial behavior so that the tissue doesn't function very well, and it can attract the immune system, which will cause then the destruction of the tissue due to the innate immune cells. So that's the bad side. The good news is there are very few senescent cells in young people, and below age 50 or 60, you don't see very many of those cells in tissues, but after about the midpoint of our lifespan, they become detectable. And now you can imagine that those cells, as they build up, they start to drive the pathologies that we associate with aging.
Probably also start to cause more, cellular senescence. And so a couple of questions sort of popped up when you were talking about sort of the double-edged sword and how cellular senescence, on the one hand, it's a function of a stress response that occurs to protect you from cancer, and so the cell stops dividing, and then the senescent cells, of course, are then making all sorts of various different secreted proteins, like you mentioned, and that sometimes it can have good effects if you're talking about wound healing and things, but also it can attract the immune system and cause, more, basically activate the immune system to make more of these hypochlorite, hydrogen peroxide, warfare things going on in the cell, which can also damage the cell even more. But the other thing is the growth factors that you mentioned that they're secreting. And, let's say as you are aging, you do have you have accumulated more damage. And let's say now that that damaged cell is, supposed to undergo senescence because that's the stress response to protect you from cancer. But in the presence of excess growth factors, that doesn't actually always happen,
No,
right?
It can start the process of cancer.
So it's kind of ironic that
Yes.
the very thing that it's protecting you from, it's also now at some point, possibly now, making you more susceptible to, at least as you get older.
So there's very good data now in mice from our lab and other labs that the presence of senescent cells can drive late life cancer, especially those cancers that are pre-malignant and poised to become cancerous, but they don't have the mutations that are needed for a full-blown cancer. But under the stimulation, the growth stimulation of a senescent cell, these cells can now start proliferating, they pick up more mutations, and then eventually they become fully malignant.
Exactly. I think I read you did some experiments out of your lab where you guys injected these
Yes.
pre-malignant cells into animals with and without the senescent cells.
That's exactly right. And with senescent cells, they converted to full-blown malignancy and, eventually kill the animal. So, you should be depressed, right? I mean, if you don't have this process, you die of cancer. If you do have this process, you're still going to get cancer. And so, we've struggled with this. We and the whole field has struggled. What do we do about this problem? And the good news is there are now mouse models. We don't know if this will work in humans, but in mice, we can selectively cause senescent cells to die. So finally, we can make them go away. And when we do that, there are health benefits. I should point out, not necessarily an extension of lifespan, but definitely an extension of health span. So our lab has done this. Our colleagues' lab at the Mayo Clinic has done this. There are now a number of labs that are working on the strategy of causing senescent cells to die. And this was initially done with transgenic mice. Of course, we can't make transgenic people, but there's an army of people looking for drugs that will do what the transgenes can do. And some prototype drugs are now, have been published in mice. They're prototypes because they're not yet ready for people, but, and there are companies that are working on these things. So the good news is, I think the strategy of tuning down the bad effects of senescence is on the way. That's on the horizon for sure. We will still have to be careful how we apply those drugs, right? If you're going in for surgery, you don't want to take drugs that will kill off senescent cells because you need to heal the wound from the surgery. So there's going to have to be some intelligent discussions about when it would be appropriate to take these drugs. The good news about the drugs, though, is that from the mouse models, it's not as though senescent cells blossom at some point after some age. They just gradually accumulate. So what that means is you don't have to take a drug that kills senescent cells every day. You may have, in mice, you can apply them every few months. And in people, maybe every few years. So this could be interesting strategy to improve health by incrementally knocking down senescent cells every so often.
Yeah. So you said that with these studies, that cleared away the senescent cells, the health span was improved, meaning I guess the tissues, the organs aged better and things like that. But I thought I remember a study, maybe it was the Mayo Clinic study, where there was a mouse model, perhaps it was an accelerated aging mouse model, where they did have a lifespan extension of like 20%.
No, so that was an increase in median lifespan.
Median lifespan, okay.
But not an increase in maximum lifespan. Okay. Sorry, and I should have distinguished between median lifespan and maximum lifespan. So the increase in maximum lifespan was not significant. The increase in median lifespan was significant. So that's what we call health span. The animals still died, but they were healthier in many respects. Mm-hmm. It's interesting. So what did they die of? It's not as easy as it might seem to determine what kills them out. Actually, even in people, sometimes, pathologists put heart stopped. Yeah.
We don't know. Yeah.
We don't know what.
Okay, so there was an increase in the median lifespan, which is still very important because now you're having more of these animals which are living longer, but they're
living healthier.
So,
and of course, for humans, this is amazing. What people dread about old age is those last years of life when, you go through a nursing home and they're barely mobile and, they can't hear and they can't see and they're depressed for good reason.
Right.
But, I think the image now of being able to be vibrant, and then who knows what will do you in the end. But, we will die. But I think what we mostly fear is those last few years of life where the quality of life is very poor.
Where you're degenerative and, a lot of these diseases that are degenerative diseases aren't necessarily like going to kill you right away, or so like you said, you could have maybe macular degeneration or sarcopenia, something where it's just slow and miserable and you can't function as well. So
and then brain age, degeneration of the brain is, dying of cancer is no fun, but I think many people fear even more this loss of cognition. It's like you lose yourself, and then the incredible burden on the family. And so the idea is to try to compress that period of morbidity so that the last years of life, you die maybe of a heart attack on the tennis court and you're winning. There's a lovely quotation from Thurgood Marshall. He was our first Black Supreme Court justice. This was in the 1960s. And someone had the nerve to ask him how long he plans to live. Lifetime appointment, right there, And he said, I plan to die at 110 from a bullet wound from a jealous husband.
That's pretty funny. But talking about the brain, you just mentioned the brain. I've always been curious about what tissues, if certain tissues accumulate more senescent cells than other tissues, including in the brain.
Yeah, so we and others have looked at senescence in the brain. Based on the markers we have, so I should also preface this by saying we don't have perfect markers for senescent cells. There are many markers, and so we tend to have some confidence that we say a cell is senescent if we look at multiple markers and we say, well, there's a good chance this is a senescent cell. So the best markers that we have been used in the brain by a number of laboratories. And it seems that the cells that are more likely to become senescent in the brain are astrocytes.
Supporting cells.
Exactly. And that's interesting from two points of view. The first is there are probably more astrocytes in your brain than neurons.
There are, absolutely.
Yeah, people don't realize that, but there are lots of astrocytes. The second is it's the astrocytes that give rise to brain cancer. So again, consistent with the idea that the stress response protects us from cancer, at least for a while. We've studied human astrocytes, and I can tell you they mount a classic senescence response, including producing all those pro-inflammatory cytokines and growth factors and proteases. And we even have new evidence that astrocytes, as you know, help protect the neurons from certain types of toxicity, like neurotransmitter toxicity. And we can show that when astrocytes become senescent, they become less effective in that protective response.
Really?
Yeah. So again, being able to get rid of those senescent astrocytes could be beneficial for preserving brain function. Now, once neurons die, that may be too late.
Yeah.
So we have to distinguish between the ability to prevent degeneration in the brain versus reversing it. I think reversal is going to be much harder.
It's always much harder, for sure.
Yeah.
Is the senescent cells that occur in the astrocytes, do you think that also, because as we age, brain inflammation also increases?
That's correct.
Do you think it's a contributing factor?
Yeah, we definitely think that's a contributing factor. It may not be the only factor, but definitely.
Well, there's now, lots of evidence showing that, a lot of cytokines in the periphery can cross the blood-brain barrier through a variety of mechanisms, including this newly discovered lymphatic system that's connected to the brain, the meninges or something.
That's right. So that's interesting. So you don't necessarily even have to have astrocyte senescence to help contribute to neurodegeneration. It could be in your liver or your skin.
Exactly. So it's sort of like just sort of all connected. Maybe we can talk a little bit about the mechanisms that lead to cellular senescence. I mean, we mentioned inflammation, you mentioned it's a stress response, it's just to stress, but, specifically...
What are those stresses?
What are the main, I know the main mechanism I always think of when I think of cellular senescence.
Just DNA damage?
DNA damage.
Yes, for sure. Anything that causes severe or persistent damage to the genome will drive cells into senescence. It makes sense because that puts you at risk for mutations. Mutation puts you at risk for cancer. So the cells want to shut that damaged cell down. But there are other stresses now that we know. We recently showed, for example, that having bad mitochondria in the absence of DNA damage, so this is just mitochondrial dysfunction, if you will, and we could show that you could cause mitochondrial dysfunction by any number of means, 5 or 6 different ways of causing the mitochondria to fail to produce the energy that they need and to produce more free radicals, even in the absence of those free radicals getting to the nucleus, causes cells to senesce. So they will senesce in response to bad mitochondria. What's interesting is the cells senesce, they stop dividing, they do start secreting molecules, but it's a different complement of secreted molecules. So we can now pretty much determine whether a cell has become senescent due to DNA damage versus bad mitochondria based on their secretory profile. There's overlap, don't get me wrong, there's overlap, but there's also distinct features that the mitochondria are responsible for, bad mitochondria are responsible for versus damaged DNA.
What's what would you say the main distinctive
So feature? One of the main distinguishing features is with DNA damage, there's a pathway that increases cytokines like IL-6, IL-8. These are very prominent pro-inflammatory cytokines. That doesn't happen with bad mitochondria. So that loop is pretty much not activated. But then other molecules are activated that can also be pro-inflammatory, but through a different pathway.
Wow. So this kind of is extremely interesting. I wonder why that is, and I wonder if there's different functions of these senescent cells. But the secretion of some of these pro-inflammatory cytokines and the attraction of immune cells to that area, one would think would then cause that senescent cell to be cleared away.
Yes, and it probably does happen, but it probably doesn't happen efficiently enough, or with age, maybe we make senescent cells at a higher rate. So several labs, not our lab, but several other labs have shown that senescent cells express molecules on their surface that can target them for being killed by the immune system. And the immune system can do that, and it does do that. Nonetheless, we still see this increase with age.
But the immune system declines with age, right? Function of the immune system. Do you think that may contribute? So maybe.
Well, that's a big unanswered question. So what happens what happens with so-called immune senescence is primarily the adaptive immune system. So
that's actually the good part of the immune
Exactly.
The quieter part.
And this is why you become more susceptible to certain types of infections with age. So the adaptive immune system tends to decline with age. The innate immune system, if anything, increases in activity with age. And it's the innate immune system that clearly targets senescent cells for clearance. So, for example, senescent cells express on their surface ligands for natural killer cells. And natural killer cells will then attack those senescent cells and kill them. Nonetheless, senescent cells still accumulate with age. So we're considering several possibilities. So one is maybe you're just making them too fast. The immune system can't keep up. The other is that although the innate immune system doesn't decline with age per se, it does change, and it could change in a way that it becomes less efficient at clearing senescent cells. So that's a big open question. We don't know the answer to that. And the third possibility is that some senescent cells may develop mechanisms to protect themselves from immune clearance, and we're sort of still studying that now, and we think maybe all possibilities are still open.
Geez, I have so many things that popped in my head that I want to talk about, like which order, because I'm going to forget. So talking about these changes in the adaptive versus the innate immune system sort of reminded me of Dr. Valter Longo's research, who I interviewed a few months ago, and he was talking about how this prolonged fasting in mice, which is about 48 hours, or translates to like 4 days in humans, which is quite a long fast, but was able to just very robustly clear away damaged cells, presumably senescent cells as well, also caused cellular death, but followed by a massive and robust increase in stem cell proliferation, sort of replenishing the population. But what was so interesting was that it seemed to, at least in aging mice, if you did this in aged mice, it normalized the difference between the innate and the adaptive. So like you were mentioning, the adaptive immune system declines with age. But this fasting sort of like replenished somehow, I guess, the adaptive, maybe some of the stem cells.
In the bone marrow, yeah.
Yeah, and so you maybe have more of a 50-50 ratio like you do when you're younger.
Right.
So what would be interesting is if you like somehow did that experiment where you did some fasting and, sort of regenerated that adaptive immune arm, I guess, of the immune system, and then like gave some chemical to cause cellular senescence and see if there's any change in...
Well, a lot of Valter's work also has to do with the side effects of chemotherapy, right? So he was able to show that with this intermittent short-term fasting, even in humans, he could not only improve the efficacy of chemotherapy, but prevent some of the side effects.
Right.
So we've shown very recently using mice, a transgenic mouse model, that some of the so-called genotoxic chemotherapies, the chemotherapies that damage DNA, definitely causes senescence. And if we eliminate those senescent cells in our transgenic mouse model, we can eliminate several of the side effects, several of the bad side effects of chemotherapy. So one possibility is that what the fasting does is it might eliminate senescent cells. More likely, what it might do, we think, is dampen mTOR activity. So just to remind you, mTOR is a kinase which is highly conserved from yeast all the way to humans. And it's a nutrient and growth factor sensing kinase. So high nutrients, high TOR activity. And what has been shown in yeast and worms and flies and mice is that if you dampen, you can't get rid of TOR activity, you need it for life, but if you dampen TOR activity, either genetically or with the drug rapamycin, which is known to target one arm of the TOR pathway, you can extend lifespan. And what we showed recently is that what rapamycin does, or dampening TOR activity does, is it also suppresses primarily the inflammatory arm of the secretory phenotype of senescent cells. So it could be that fasting and rapamycin and the secretory phenotype of senescent cells all come together around the TOR pathway, and that it's really TOR activity that's driving both the aging phenotypes, the side effects of chemotherapy, and may partly explain the benefits of the short-term fasting that Valter is a proponent of.
Interesting. So if it's dampening the secretion of these cytokines from the senescent cells, but it's not actually getting rid of the senescent cells, or...
It's not, so far as, well, we've shown that either genetically or pharmacologically with drugs like rapamycin, suppresses the secretion of senescent cells, but it doesn't kill them. So unlike some of these other drugs, that these so-called senolytic drugs that actually kill senescent cells, the mTOR drugs, the mTOR dampening drugs, suppress the ability of the senescent cells to secrete. And the effects are last longer than the application of the drug, in the sense that we know that part of that secretory, pro-inflammatory secretory phenotype is due to a feedback loop. And what dampening mTOR does is it breaks the loop. And the loop takes time to reestablish. So even after you withdraw the drug, you still get suppression until eventually the loop reestablishes and then the cell starts secreting again. So you might think that if you were to fast for 4 days, every few weeks, you might have more benefit, certainly more benefit than taking a drug like rapamycin, which has side effects.
Right. Because you're not only, when you're fasting, you're also clearing away the damaged cells, in theory. I mean, we don't know how much of that's occurring in humans yet,
yes,
but we do know
in mice,
that's right. That happens. And then the other thing, when it kind of goes back to the mitochondria-induced senescence that we talked about a minute ago, is that we know that fasting increases NAD levels, and so the NAD plus NADH ratio. And that sort of is very interesting because this mitochondrial-induced damage, I think, has something to do with declining NADs.
It does, actually. So that's what we've shown, is that this mitochondrial dysfunction-induced senescence, we call it Mitochondrial Dysfunction-Associated Senescence, or MIDAS. So we call it the MIDAS phenotype. Really has to do with this altered NAD-NADH ratio, and that's one of the drivers. Interestingly, so when you change that ratio, you activate a kinase called AMP kinase. AMP kinase is a major regulator of P53. P53 is a major regulator of both senescence and apoptosis. So that could be the link of why, in Volta's paradigm, you get reduced inflammation. That could be due to suppressing the secretory phenotype, but also increased apoptosis because you now have activated P53.
Right, exactly.
Yeah.
So I just got sort of lost in the question I was going to ask you, but okay.
No, one other thing I was going to I don't know if you're going to ask this question, but it's worth discussing, is the stem cell, the regenerative process that Volta has seen. So we also know that the secretions of senescent cells can have very profound effects on stem cell proliferation and function. So it could also be that by dampening the secretory phenotype of senescent cells, you now release those stem cells from the suppression that was due to those secretory phenotypes, and therefore allow them now to do what they do best, which is to proliferate and regenerate a tissue. So all of these things really probably tie in to each other. I mean, they're all interrelated.
So the growth factors that are actually secreted by the senescent cells do help with stem cell growth? Because I always thought of senescent cells like, if this happens in a stem cell, you're depleting the stem cell pool and it's contributing to stem cell aging.
Both are probably true.
Okay.
Both are probably true. So we have shown in the skin, for example, that with age, senescent cells do accumulate. But if you clear those cells, you don't get much benefit. And that's because by old age, you've depleted the stem cells. So, you can't, once you've depleted that stem cell pool, you can't go backwards, or at least you can't very easily go backwards. But 2 labs have now shown that senescent cells can also produce growth factors, or factors, that help neighboring cells reprogram to stimulate regeneration. And they do it, again, by their secretory phenotype. So it's, again, this double-edged sword. Some of the secretions of senescent cells dampen stem cell activity, and others promote stem cell activity. So
what about the mitochondrial-induced senescent cells? What's their function? I know the DNA damage-induced ones are, obviously are protecting from cancer. What Like, what's the purpose?
What is the evolutionary purpose? Well, if you have a cell with bad mitochondria, you probably want to clear that cell, prevent that cell from propagating, because then you're going to have clones of cells with bad mitochondria. And we know that that causes all sorts of degenerative diseases, neurodegeneration, as well as muscle degeneration. And so it probably is also protective, but not so much against cancer, but against accumulating degeneration within a tissue. We know, for example, that people who are born with mitochondrial DNA defects, eventually the bad mitochondria expand. And so that's not good for an organism. So there probably is a protective mechanism to prevent the propagation of cells with bad mitochondria.
Okay, well, that makes more sense.
Yeah.
I guess, at a certain point, your mitochondria, if you don't have, a really bad defect in mitochondrial DNA, your mitochondria will repair themselves through mitochondrial fusion. And right? Isn't fusion also part of how they sort of exchange all their mitochondrial content, and the damaged one sort of fixes itself to some degree? Although I guess that gets diluted.
The bad ones also get eaten up by the lysosomes. Yeah.
Right. Yeah. So I guess there's multiple mechanisms. Do the mitochondrial-induced senescent cells, they produce the growth factors that affect stem cell growth?
They produce some growth factors, yes. They produce, for example, amphiregulin, which is a EGF-like growth factor. So they do. So we're still struggling with trying to understand, what aging phenotypes are caused by genomic damage, what aging phenotypes are caused by mitochondria.
Muscle atrophy, do you find more in muscle tissue, mitochondrial induced?
Well, we find more senescent cells. We don't know, and we find them in the heart, but we don't know how they got there. It's a big unanswered question as to when you see a senescent cell in vivo, which we do in human tissue and mouse tissue with age, they accumulate, but what caused them to become senescent? Is it mitochondrial damage? Is it genomic damage? Is it metabolic imbalances?
Right.
We really don't know yet. It's still one of the big unanswered questions.
Yeah, that sort of leads me into the preventative sort of questions I had, and that is, we do know that there are lifestyle factors that affect mitochondrial health, that affect DNA damage, that affect telomere length. And these things are obvious to a lot of people. I mean, a lot of work out of Elizabeth Blackburn's lab with Alyssa Appel.
Yes.
They've done some really great work. I mean, a lot of it's associative studies where they're looking at telomere length and various lifestyle factors, but they've shown, for example, that exercise is very important, and people that are sedentary have shorter telomeres than people that are physically active. People that are stressed have shorter telomeres. Actually, people that have low vitamin D have shorter telomeres. Omega-3, sugar accelerates the aging process at the level of telomeres. So all these factors, and telomere length also is a major regulator of cellular senescence.
Oh,
right?
yeah. I think the best way to think about telomere length is that when the telomeres become too short, so telomeres form a structure that caps the ends of the chromosomes and makes sure that the cell doesn't mistake that chromosome end for broken DNA, because the cell will try to fix that broken DNA by fusing it to another broken end. And you don't want that to happen with your telomeres. So when the telomeres become too short, that cap structure then is disrupted. And now the cell thinks it has a broken DNA end. It will try to fix it if it can, but if it can't, it will cause senescence. So short telomeres are in a way a subset of DNA damage, which again is a major driver of senescence. So telomeres tie in.
Right.
Exactly.
They're so connected. Yeah. And don't telomeres sort of take the hit for DNA damage as well?
Well, they have a fairly high proportion of the nucleotide guanosine, right? And that base is pretty susceptible to oxidative damage. Exactly. So it becomes like a sensor for damage. It's not the only thing that will cause DNA damage, that is oxidized guanine, but telomeres are are good sensors of whether there's oxidative damage.
So do you agree that things that at least have been associated with DNA damage and telomere length in humans in associative studies may likely be the same things that help prevent the accumulation of cellular senescence?
Yes,
to some degree.
Yeah, to some degree, exactly.
Right.
I mean, I don't want to oversell the idea that senescence can explain all of aging, just like telomeres can't or DNA damage can't. But we think it's an important process, and it's tied in to other things that may intersect with the senescence pathway, including things like telomere length and genomic damage, but also things like exercise. So there's a recent study that I thought was really interesting. This was a study that looked at lifespan, longevity in obese people. So both groups were obese, but they compared obese people who were sedentary with obese people who were moderate exercisers. And there was something like an 8-year difference in lifespan.
You're kidding.
No. Even with the obesity, moderate exercise in general protected the longevity of obese people. And so imagine what it does for people who are not obese. I think exercise is probably the single most important intervention that cuts across multiple diseases. So sarcopenia, which is, muscle loss with age, a major cause of, you see people sitting in wheelchairs like this, it's horrible. The only effective intervention is exercise. Mm-hmm. So and we know that exercise can have some effects on senescent cells in vivo, but we don't know how it works. We don't know precisely what it does.
What about the stress response pathways it activates? I mean, exercise is a type of hormetic stress.
That's right.
Right, so it's a little bit stressful and it's activating all these.
Yeah, so that's one idea, is that it's hormetic stress, meaning it's low-level stress that then primes everybody else, all your other stress responses, to be hypervigilant. Mm-hmm. And so then when a bad telomere comes along, or an insult from radiation, or high sugar because you just couldn't resist that last brownie, you're better able to deal with that stress. It's one hypothesis. I think there's still, an ocean of ignorance around how exercise seems to be so beneficial for so many indications of aging. Again, aging, not necessarily maximum lifespan, healthspan. Mm-hmm.
Yeah, it does seem to affect many different diseases of age as well, in addition to sarcopenia. Cardiovascular health, I mean, cardiorespiratory fitness is also very tightly correlated with.
And there are some groups now that are studying the effects of exercise on side effects of chemotherapy. Right. And showing benefits of, again, mitigating some of those side effects simply by an exercise regimen. And we're not talking running marathons. We're talking sort of moderate but persistent exercise. I think it's
fascinating. I've read a couple of the studies, at least the animal studies, where they can sort of force them to run a little bit more on this
running
treadmill.
Wheel. Yeah,
and it was like there was a very robust response in terms of, at least in combination, I think, with standard of care treatment.
That's right.
I want to say something like 50%. It was something very really, that's very. I've always thought about it as sort of when you're exercising, you're forcing your mitochondria to work harder and you're producing more reactive oxygen species. And cancer cells don't like that. I mean, they're, so who knows?
Yeah.
Who knows?
Yeah, I think, mechanism so far unknown, but lots of possibilities.
Right. What about the hope for a clinical assay for measuring things like cellular senescence, or at the very least, DNA damage in people?
Yeah, so that can be done. I mean, I know of at least a couple of companies that are doing this. Usually they use peripheral blood, and there are very good antibodies that will detect persistent DNA damage. So you can stain these blood cells and get a sense of what your DNA damage load is. There are good markers for senescent cells now. We always recommend that you use 2 or 3, but we can probably assess the load of senescent cells. The difficulty lies in tissue specificity. Mm-hmm. So peripheral blood is easy. Buccal swabs are easy because they're accessible, but you really probably want to know how many senescent astrocytes you have in your brain, and brain biopsies are not going to be approved very soon. So that's the difficulty. We can get a general idea from those easy to access tissues, but there are tissues we might want to know about that are not going to be easy to biopsy.
Is there a correlation between, let's say, if you were to look at cellular senescence in white blood cells or leukocytes, between that and the heart or the brain? Or
Yes, there is, and of course the work that really exemplifies that the most is the work on telomere length, where you take peripheral blood. That's mostly what's being used now to assess telomere length. But you look at the data. And of course, there's enormous scatter in the data. There's always a young person, who's down with a length that's equivalent to a 90-year-old, and a 90-year-old who's up there with the equivalent of a 16-year-old. And part of that, I'm guessing, is due to the fact that you're measuring one tissue and you don't know what the history of that, when was the last time you had a cold? Right. That's going to affect, how many T cells you have with bad telomeres or not. And so human data tends to be messy.
It's very messy. Yeah. I've actually seen this quite a bit because I've done a lot of work with Dr. Bruce Ames, and I've measured DNA damage in people, in lean people, obese people, after a certain, giving them a certain intervention. And I do this by measuring phosphorylated H2AX, gamma H2AX, right?
Yes.
But I've seen, even looking at different age groups, like, sometimes like the 20-year-olds will look like a 70-year-old, and you're like, what happened, here? Or sometimes the obese people look really great, but most of the time obese have higher levels than lean. But there's certainly, there's a lot of variation. There's a lot of variation.
And I think that variation is twofold. The first is, we're not genetically identical the way our mice are. So there's going to be individual to individual variation because we're not genetically identical. But the other interesting aspect is that we've taken, in our mice, we have transgenic mice in which senescent cells activate a protein, a luciferase, that we can then measure by luminescence in the whole animal. Mm-hmm. So we can follow the appearance of senescent cells in living animals by looking at this luminescence signal. So we start with, say, 12-month-old mice. So that's a 35 or so year old person. Very low signal. And then as these animals age, the luciferase signal goes up and up and up. Then you look at the error bars. Genetically identical animals, sometimes in the same cage, and the error bars get larger and larger and larger. So that says there is stochastic variation that's not due to genetic differences that causes identical animals to have some with a high burden of senescent cells, some with a low burden of senescent cells. And this is true for virtually every aging marker that has been looked at. The error bars get larger and larger and larger. So we call this stochastic variation. We don't know whether it's malleable, meaning we don't know whether you can make things less variable or more variable. We don't know whether it correlates with health of the animals, but it is a very common feature of aging is that things that go wrong do it almost randomly.
And there's no idea of what's causing it? It's just...
Well, I mean, you know that if you take a bunch of cells, identical cells, genetically identical cells, and you apply some toxin, and then you look at damage, you get a Gaussian curve, meaning there are some cells that don't respond very well. Most of the cells respond a certain way, and then some cells that are super responders. And so it probably is just the messy nature of biology. We have all these pathways that are intertwined, and by chance, one configuration makes the cells super responsive, and another configuration makes a cell less responsive. And if that's true of cells, then it's definitely going to be true of something as complicated as a mouse, much less a person.
Right. So I have just a couple more questions, a couple more wacky, but just to circle back since I have you here, I know we've been talking for a while, but back to the NAD levels, that reminded me of this whole new field that's sort of semi-new, I guess, on, different precursors of NAD that you can give, like nicotinamide riboside and nicotinamide mononucleotide. I know nicotinamide riboside, at least in humans, has been shown to increase NAD levels, at least at very high levels.
Yes.
What do you think? Do you think that any of those, and I know that there's been some animal studies showing it increased health span so that the tissues were aging better and certain organs were aging better. Do you think that any of those effects of the increased NAD had to do with lower cellular senescence?
That's a good question. As I mentioned, we know that this mitochondrially driven senescence is definitely driven by this alteration in the NAD-NADH ratio. So it's possible. We haven't really studied those precursors directly on senescent cells, but
it'd be interesting.
It sounds like a grant I should.
Yeah, right. There you go. And then one more question before I get to the other wacky question was, what do you think about some of these like, people call them quote unquote fasting mimetics? I don't like that. I think there's too many things going on with fasting, but that have been shown to clear away damaged cells, like spermidine or the hydroxycitrate or resveratrol.
Yeah.
Do you think that's something? I mean, people are taking these to like clear away senescent cells.
Yeah, we've explored some of those. Some of them have no effect on senescent cells, which doesn't mean that they might not have health benefits, but it just doesn't act through senescence. Resveratrol, for example, we don't see any effects on, say, pro-inflammatory cytokine secretion or anything that we think might be important, but that doesn't mean that it's not doing other things. Okay. Yeah. I much prefer red wine. Cells don't like it.
Yeah, cells don't like it. The other wacky question that just came to my mind was, so I'm thinking about cellular senescence, not the mitochondrial senescence, but the DNA damage to induce cellular senescence as a protective effect against cancer, and that's why it's evolved. I mean, we have that because we don't want to die of cancer young. What about animals like elephants that don't get cancer? They have a relatively long...
I don't think it's that they don't get cancer. I think what's amazing is that they're so big, right? And they have so many cells.
Yes.
And so you would think that there should be super ways they have of protecting against cancer, because it's a hell of a lot of cell division to go from a single elephant egg and sperm, a zygote, to an elephant, which is so big. And there have been some studies on looking at, for example, tumor suppressor mechanisms in some of these animals. P53, right? They have extra copies of P53. And so...
But do they have any cellular senescence? I was just wondering if anyone's looked at that.
I don't know that anyone has looked, but I would be surprised if they don't. Mm-hmm. I mean, we've seen cellular senescence or we, meaning the field, has looked in a number of vertebrate species, and it seems to be common amongst all vertebrate species.
Does it occur in lower organisms? They only live...
Well, it does. Based on the markers we have, some people have looked in C. elegans, and they don't seem to find it there. But then C. elegans is unusual in that the only dividing cells in the worm is the germline.
Right.
But in Drosophila, there is a small fraction of cells that undergo division in the gut. And there is some hints that there may be senescence that occurs in the gut of the fly. And that kind of makes sense, because one thing that happens with age in the fly is they get this gut hyperplasia. It's almost like colon cancer in the fly. But they don't have the vast number of dividing cells that, say, we have or a mouse has. So it's possible that it might be found in Drosophila.
Okay. And what about that? Just now, one triggered a question. So does cellular senescence occur in more rapidly proliferating cells? Is that like the gut in the human?
It can. I mean, the gut is different I think, because remember, those cells are programmed to die.
Right. Kind of like the immune system, I guess.
Or some immune cells.
Yeah.
I mean, but if you think about, say, something like the skin, so again, those cells are programmed to die, but then there are these basal epithelial cells. It takes a long time. So unlike the gut, where there's very rapid sloughing off of those cells, it takes longer in the skin. And of course, people do get skin cancer, and they always come from the basal keratinocytes. This is certain types of skin cancer, right? Not all skin, I mean, not melanoma, for example, which comes from melanocytes. But so those cells can transform even though they're programmed to die. And we do see senescence in the basal layer of keratinocytes in human skin. So maybe.
Does that contribute to collagen breakdown?
Well, that's the idea, is that it could, because they're making a lot of proteases
right.
that will destroy collagen. Yeah. But I mean, we have, nobody really knows. Yeah.
Okay. And so with all your research that you found, throughout the years on cellular senescence and just aging in general, do you have any practices that you have sort of gleaned from your own research that you incorporate into your own lifestyle?
Yeah. So, moderate exercise, although I always seem to be busier than I like to be. A good diet, I don't smoke.
Lots of greens.
Eat your veggies. But then there's this genetic component which says you should choose your grandparents very wisely. And,
and that's sort of difficult to do. So, well, Judy, this has been a very illuminating conversation. I've really enjoyed it.
Thank you for your interest.
Very interesting. And I look forward
the audience for their
to looking.
interest.
I'm going to continue reading about your research. I can't wait to learn about all the new things that you discover. But for people that want to learn more about your research, do you have a website or just the Buck Institute?
Yes, they can go to the Buck Institute website, and there's websites for the individual faculty members.
Right. I know if you just Google Judy Campisi, the Buck Institute's like the top hit, so they can learn more about your research there.
Yes, of course.
Okay, well, thank you, Judy.
Thank you. Thank you.
If you enjoyed this episode, make sure to click like and to subscribe to the channel. You can also subscribe to the iTunes podcast. We are on there as well. Finally, if you like the channel, you can help support it by becoming a direct Pay What You Can subscriber by heading over to foundmyfitness.com/crowd sponsor. Thanks for listening.